Any questions? Yeah.
So is myotonia similar to dystonia?
I'd have to look up that definition of that word. I don't know if that word describes people with myotonia or not, honestly. Okay, so here is averaged graphs from a normal person and periodic paralysis. So you can see that there's stochastic noise, but when you average a normal person, you're seeing depolarization and back up to baseline. In periodic paralysis, you see depolarization and it doesn't get back to baseline. And so basically, you've discharged your Tesla battery and you're not charging it back up enough to discharge it again. And so you can't fire it again. You can't flex again. And so that's why your muscles are lax, okay? So the main way that you'll see that on the boards is probably a sodium channel mutation. And so, well, I won't say the main way. You could see this definitely both on boards and with my stuff. So the sodium channel can become leaky. So remember, you're storing up energy as a battery, high sodium outside, high potassium inside. And so the leaky sodium channel is allowing that sodium to come in, okay? And as this presents, it can last minutes to hours, okay? You can also have periodic paralysis, and it's to me, it seems like a very narrow range that this actually happens, where you can be either hypokalemic or hyperkalemic, okay? And so you'll have to find this out patient by patient. You'll probably, I don't know if you'd end up sequencing the channel to see what their defect is. You probably would at this stage. I honestly don't know that. But you would treat them a little different. So if they are hypokalemic, that's measuring the concentration of Ka potassium. If you're measuring the concentration outside the cell, right? So hypo is low potassium outside the cell. Hyperkalemic is high potassium outside the cell. So if someone is hyperkalemic, high potassium outside the cell, and they have fruit juice, which fruit's high in potassium, that can hurt them because you're adding extra potassium, okay? That's kind of a good board question because it's, you know, not the norm for, you know, fruit's bad in that case. Okay, so for myotonic dystrophy, the abbreviation's DM just because of the verbiage of how it was termed. Here's a normal graph for myotonia. Sorry, a lot of feedback. You see these repeated spikes. And then this graph on the right is showing a normal cell, but where chloride is replaced with some ion that can't go through the channel, and you see it looks like myotonia. So it's recreating the graph phenotype of where the chloride's not getting through the channel, right? So there's a bunch of different mutations. So the gene CLCN1 codes for this chloride channel, the protein CLC1, and that helps repolarization. There's a bunch of mutations that basically hurt that, okay? So you decrease repolarization. I think this can be a little confusing to people. The genetic link to myotonic dystrophy isn't totally understood. We kind of just get a high level of what we understand about it from me. Ready? There's two genes that are involved. The first is DMPK. It doesn't matter what it does. I think I have that in the notes, and I'll tell you why. The main thing about DMPK is that it has a trinucleotide repeat, and so it has that anticipation. The more repeats you have, the more likely you're likely to have myotonic dystrophy, and the worse it's likely to be, okay? DM2, myotonic dystrophy type 2, is caused, it's a lesser form, is caused by a tetrarepeat, CCTG, in a different protein, CNBP, okay? That does not have anticipation or as much anticipation as DM1, okay? So you'll definitely, they won't ask an anticipation question about DM2, it'll be DM1. And I'll show you data about that in a second. Now what's interesting is the way that these affect the chloride channel is that these repeats, okay, so you have a protein, now you're having a lot of extra DNA. That DNA becomes RNA and then hydrogen bonds back on itself and forms this big structure, and it binds other proteins that bind RNA. Those proteins that bind our RNA normally splice CLC1. And so you're kind of taking away all the machinery that CLC1 needs to splice correctly. And so CLC1 splices incorrectly because these other proteins are binding those splice protein, or sorry, these other genes, mRNAs, are binding that splice protein, okay? Does everyone understand that? It's a weird off-site effect. And you can imagine in different cells, weird repeats like this could be affecting other splice proteins. It's also worthy to think about what kind of drug targets that creates, right? You could try to not adjust any of this protein expression, but try to upregulate the proteins that are doing the RNA splicing to compensate, right? Okay. So here's some data showing that anticipation and the trinucleotide repeats. So here is the CTG size. So the higher up, the more repeats, okay? And you see that the more there are, you have worse versions of DM1. So congenital, then infantile, then juvenile, then adult, then late onset. The ones to the right here, late onset adult, being less severe as well, okay? Here, on the bottom left graph, we have the x-axis is age at onset, and then the different colored bars are these same binned types of DM, so congenital, infantile, juvenile, adult, and late onset. And then you see the effect that they cause, deleterious effects. So cataract surgery, pacemaker, cataracts. So basically everything on the Y is bad, and you see that congenital, the thing that happens more when you have more repeats, all these things happen earlier when you have larger repeats, right? So all the colors get shifted earlier and earlier. So you don't really need to see this data. I just wanted it to fire neurons for you to believe that more repeats not only say they're going to get the disease more likely, but that the disease will be worse. So I think in the next 10 years, this will start happening, that people that have IVF might select, if I was a parent or a to-be parent that had trinucleotide repeats that were not yet manifesting but were close, you could imagine selecting embryos where it didn't go over that line, right? Whatever, I'll go off a little bit on a tangent. People talk about, there's a lot of scaremongering about like genetic medicine and what it might do. One of the kind of things that may happen is that we may lose resilience kind of in a biodiversity sense. This is just totally sage arbor pontificating, no background, right? But you can imagine if a lot of people started doing this, the percentage of population that had CTG repeats that were close to but not manifesting would go up, right? Because the sad way of saying the flip side was those parents would have had a child that had more repeats and either died or didn't procreate, and so you kind of prune the tree there, right?